Everyone was thinking about that.
I was just thinking, like, is there, how quickly can this happen? And you have to like, get all of these like kind of out, but there's only so many that are actually paid for.
Apparently very quickly. I can't remember the number that, I was reading another book just to get a different perspective this morning. And while I can't remember the number that the book quoted, something like 10 to the 21st molecules of ATP that we make a day. But in terms of recycling ATP, now remember, we're recycling the same molecules. When we say we're synthesizing ATP, we're making ATP, then doing something so that it becomes ADP, and then we're remaking ATP. So we're cycling the same small number of molecules. But over the course of a day, if you were synthesizing all that ATP new, it would add up to approximately equal to your body weight. So, this is all happening very, very quickly. But in part it can happen very, very quickly because of the small spaces we're talking about. The distances these things are moving are very small, right? We can create fairly substantial gradients over a fairly small distance. So these things can move and then move back very quickly so that we can keep this cycle going. Okay? So, we've talked about difference in concentration being a difference in energy, right? But also we have a separation of charge. So just like when you talk about the ion gradient, the electrical potential created in nerve cells when you create the sodium and potassium ion gradients for nerve cell transmission, for action potential transmission in physiology. You haven't taken physiology yet? You'll be amazed when you talk about physiology. You'll talk about all that stuff, right? Well, in physiology, you talk about differences in charge creating an electrical potential, right? It's really why we talk about oxidation and reduction in general chemistry. We're talking about things we learn in general chemistry that help us understand biological systems. So not only are we creating a difference in concentration, we're creating a separation of charge, which creates an electrical potential. So out of this, we get chemical potential energy plus electrical potential across that membrane. And so the total energy stored in creating this gradient is a combination of the chemical difference in concentration and the electrical potential difference created because we have created a situation where we have more positive charge on the outside of the membrane than we have on the inside of the membrane. So we have a difference in charge and a difference in concentration. That creates two forms of potential energy. Okay? So, Robbie said that the whole point of this was creating this difference in concentration, this gradient. Well, this gradient is a form of energy, right? It's creating energy in two ways, which means, right, the whole conservation of energy thing, where did we get the energy to create that energy difference?
NADH.
NADH, right? So NADH, and quantitatively to a lesser extent, FADH2, those are high energy molecules. Their oxidation releases energy. That energy, some of that energy is stored as this difference in potential. Where does the rest of the energy end up? What happens to the rest of the energy? No. Lost as heat, right? So, we lose some of this energy as heat. Right? So this storage process is not 100% efficient, so some of this is lost as heat. Now, what do we want to happen next? What are we going to do with this, right? Well, let's go back a step. Where did this energy start? If these were high energy molecules, where did we get the energy to make these molecules? What? Well, specifically, but doing what? Where was the energy before? What was the citric acid cycle doing? Well, it doesn't, again, right, we have to go back and think about the law of conservation of energy. Where was that energy before? No, not ATP. What was the citric acid cycle doing? It was making NADH by doing what? What? Be more specific than breaking down. What is it actually doing to acetyl-CoA?
Oxidizing.
Oxidizing, right? So this started out as energy stored in the bonds of organic molecules, right? Right, in different forms. Acetyl groups, succinate, glyceraldehyde phosphate, right? We've had all sorts of sources of organic molecules that were oxidized to make either NADH or FADH2. We're going to add to that fatty acids. If we have time, we'll talk about some specific amino acids, right? So oxidation of a number of organic molecules, right? So these organic molecules must have been high energy. We don't think of them that way, but we were able to oxidize those to store electron energy in NADH and FADH2. We were able to use that energy then to create this hydrogen ion gradient. Now what are we going to do with the energy of that gradient? Now, Sarah, we're going to use it to make ATP. It's ultimately that difference in energy that we have created that we're going to use to make ATP. So are there any questions about what's going on in the electron transport process?
I have a question.
Yes, ma'am.
Okay, before, like at the end of the last class, we started to talk about a mechanism with tyrosine, like in copper or something.
Ah.
And I didn't know if that was something that you thought was important.
Which one was it?
I don't know.
Big circular slide.
Big circular slide. Not that one. Not that one. That one?
Yep.
No, I didn't want to do that.
Okay, just making sure.
I decided it was going to give me a headache.
Okay.
Okay? So back to here, right?
Oh.
What's the matter? Why is that a problem? Okay, so we've transported our electrons. I will tell you, we're going to encounter some other, right? Especially when we talk about fatty acids and amino acids. We will talk about other processes that direct electrons into the cycle by reducing coenzyme Q, right? The succinate dehydrogenase is not the only one that introduces electrons at that stage. So now what do we have to do? We have to make some ATP. Okay. And so we get to the ATP synthase complex. the ATP synthase complex uses this gradient of hydrogen ions, right? And hydrogen ions coming back into the matrix through the pore portion of the ATP synthase drive, electrons drive. Yes, I got my tense proper. And I messed up my proper tense. Right? The hydrogen ions drive formation of ATP. Okay? Now, this system is actually kind of cool. If you look at this, you can actually see the little bulges of the ATP synthase complex. It looks like little mushrooms on the mitochondrial inner membrane. So this is an electron micrograph. Those little things right there, those are the ATP synthase complex, the mushroom head of the ATP synthase complex, extending into the mitochondrial matrix. So you can actually see them. And what's interesting is, standing here close, it looks all sort of gray and grainy, but I can actually see it more clearly if I look on the screen of the iPad at a distance. You can sort of see them. They look, it's like the resolution gets better. It's too pixelated because I'm too close to it here. So you can actually see them. Right? They physically look like a mushroom with a stem off the side, and then this is the membrane associated portion. So this is sticking into the matrix, this part's in the membrane. Okay? This is just the globular head portion. It's composed of alpha and beta subunits. Right? So this is the part sticking into the matrix. This is a space-filling model. Great, great, great. This is looking down the core. This is showing essentially the whole thing, a model of the whole thing. So this is embedded in the membrane, right? There's this stem, which is a gamma subunit, and then there's this globular head, which is a trimer of dimers. So they're alpha-beta dimers, 3 of those dimers together in a trimer, structurally. Okay? So you've got a helical portion that is membrane associated. You've got this portion, and then you've got the alpha-beta subunit. This is where ATP is actually synthesized. Now, let's see, do I have the picture that I want? I hope so. Okay, so to try to figure out how this thing works, someone reconstituted this. By putting a biotin on the external part of the C subunit, right? So this is on the, then the biotin, there's this protein streptavidin, which binds specifically to biotin. It's a biotin scavenging protein. But in this case, they're just using the streptavidin to bind to the biotin. And attached to the streptavidin is an actin filament.